think we'll get started. I'd like to welcome you all to this webinar series on promoting animal health and welfare via pathogens control. I'm Laura Boyle. I'm the president of the EAAP Commission on Animal Health and Welfare, and we're hosting this session today. So it's the 17th EAAP webinar. They've been running for almost 2 years. And they typically run every month on the second Tuesday, and the purpose is to inform, update and disseminate innovations from high-profile speakers to all our individual members in the EAAP. So as I said, today's webinar is being hosted by the Animal Health and Welfare Commission, and we in our commission, our aim is to support scientific debate on animal health and welfare across all livestock species. Obviously, animal health and welfare is a very multidisciplinary field, and this is very much reflected in the high degree of collaboration we have with the other species-specific study commissions, so the Cattle Commission, the Pig Commission, and with these commissions, we typically co-host a lot of scientific sessions during our annual meetings of the EAAP. And we also have the Animal Behaviour Working Group of the EAAP is affiliated to the Animal Health and Welfare Commission. So more specifically onto today's proceedings, we want to thank Standard Biotools, who are the sponsors of this webinar, and just a few words on Standard Biotools. They're formerly known as Fluidigm, and they develop and manufacture genomic platforms, and the agricultural genomics field is responsible for a lot of breakthroughs in the health and performance of crops and of animals. They've yielded They have contributed to increased yield, pathogen resistance, and stress tolerance. And the main, I suppose, work of Standard Biotools is they have proprietary technology based on these microfluidics, and these typically minimize the cost of labor and material and provide highly reproducible data from robust instrumentation for pathogen detection, breeding programs, and animal health. So the lineup today then of this session, which is about promoting animal health and welfare via pathogens control, we have myself speaking for the first few minutes, and I won't take up too much of your time. I'm nearly done, and I'm going to pass you then to Christian de Croo from INRA, who's going to talk, whose title of his talk is specifically the title of the webinar, Animal Health and Welfare via Pathogens Control. And then we will move to Nicole Gulka from the University of Copenhagen in Denmark, and she's a postdoc there, and she's going to talk about the design and validation of high throughput real-time PCR systems for detection of porcine and bovine respiratory and enteric pathogens. And then we'll have some time to wrap up and some time for questions and answers. So on that topic, what I'd like you to do is, if you have any questions that come to mind during either of the talks, you can raise your questions in the chat box. And if you could indicate, please, as to who you want them directed to, if you want them directed to someone in specific. This webinar will be recorded and further and available in the future on the EAAP website, members restricted area, and on the official EAAP YouTube channel. So you should stay tuned to the EAAP website to find out about forthcoming webinars, and we also publish them on social media. And we do have some preliminary information on our next EAAP webinar, which is a nice title, Work Like an Ant, Collaborate Like a Bee, Insect Research Collaboration Initiatives in Europe. And that's going to be on the 14th of March. But, you know, as I say, follow the website for more information on that. So I will pass you now to our first speaker. I'm just going to stop sharing my screen. I don't think I have stopped sharing my screen. Someone wants to notify me.
You're fine, Laura. We can move to Christian.
Okay, yep. So Christian, if you'd like. So Christian de Croo is a senior researcher at INRA France and deputy head of the Animal Health Division. He's been working as an epidemiologist, as an epidemiologist. So I'll pass you over to Christian now. Sorry, Christian.
Thank you, Laura. And thank you, Andrea, for inviting me to give that talk. So a few words. So the title of the talk, in fact, it was provided by Andrea. and like a comment, in fact. And it's an opportunity for me to address that question that is not so simple, in fact. And I will give you a point of view of a non-specialist of these issues, because it's very multifactorial, multidimensional, multidisciplinary. So it's not easy to be a specialist of everything. But I am a very strong, interest in this question. And just to explain in a few words where I'm come from, just to explain my point of view. So I moved from veterinary practice to research in the field of epidemiology. So I had a short period of veterinary practitioner in dairy cattle, beef cattle, and sheep. And then I moved to the field of epidemiology in research. I worked on different diseases, and I had a collaboration with sociologists for about more than 10 years on the decreased use of antimicrobials. And currently, I am a senior researcher in the ASTRE research unit at CIRAD, that is based in Montpellier, in the south of France. And this research unit works on the integrated approach for animal health, including both biotechnic aspects and sociological aspects, and also working on disease surveillance and control. And apart from that, that work, I am at INRAE. I am also in charge of the program on animal health and welfare. I want to tell you a couple of words about that, because it's exactly in the topic of today. So this interdisciplinary research program at INRAE is called SAMBA, and it regards animal health and welfare of farm animals. And the goal is to generate knowledge that will enable changes to livestock practices and production systems, so as to guarantee both health and welfare of animals through their lives. And within that program, there are various studies considering animal health and welfare jointly at various scales: animal, farm, animal sector, and also the link to the society. And it involves veterinary and animal sciences, but also human sciences. And it's a 5-year program that started 3 years ago. And we are funding projects based on different goals. But this is internal to INRAE. And now I move, if it works, and I move to the question that was asked: does pathogen control have a positive impact on health and welfare? And I will provide a point of view from the veterinary and animal sciences first, for the most part of the talk, and then a short period on human and social sciences point of view. So if we start with veterinary and animal sciences point of view, in fact, it's a very old story, and because it's the traditional approach of preserving animal health is to fight against the pathogens and have pathogen control. And this was done for animal diseases that are very important economically, but also frequently to preserve human health. And even 2.5 centuries ago, during the fight against rinderpest in France, it was the occasion to create the first veterinary school in the world in 1762. So in fact, the idea is to preserve animals from pathogens with different means. The main important ones for me: animals kept indoor, in cages, on slatted floor. So this is done to control various environmental factors, including infectious disease and parasitic diseases. And for example, we got rid of avian tuberculosis and various parasites when we put poultry in flocks with intensive farming poultry. And also a counter example is the return of chicken red mice with the possibility of outdoor access these days. The second means the biosecurity measures to prevent contact with infectious threats, and the important current example is highly pathogen avian flu that can be zoonotic, and the idea is to keep animals indoor, and not the idea is the regulation. If this is mandatory to keep animals indoor in infected area. And the third mean is I put that in a control. I put that also in the same topic, the aspect of vaccination. It's maybe a bit too large, but I think we can put that here. So it's vaccination and also the eradication plans that are built to control specific infectious diseases. And for example, we vaccinated and then later we eradicated foot and mouth disease in the EU in the 80s, or the example of stamping out of infected cattle to control bovine TB, that is zoonotic, and since the 50s. So this is an old story, but it was adapted to new other topics. And for example, recently, just to decrease the need of antimicrobials, many veterinarians use just a program against pathogens like vaccination against, for example, colibacillosis for post-weaning piglets, or use of auto vaccines for different pathogens, Pastorella, Streptococcus, Staphylococcus for different species, and also different eradication plans of viral diseases to control, to decrease the bacterial complications, and so to decrease the need for antimicrobials. So just to summarize, the control of pathogens is one of the main preventive means that are used by veterinarians to preserve health at the farm level and also at the national level with regulation. Other means are more focused on better environmental conditions, better nutrition to reinforce resistance and immunity. So if we move to welfare, in fact, in my understanding, pathogen control was never done for welfare conditions in the first instance. And if we start from the definition of animal welfare, that means the physical and mental state of an animal in relation to the conditions in which it lives and dies. In fact, good health is one of the components of welfare, and any disease, including pain, suffering, decreased physical possibilities, has a negative impact on welfare. So in that sense, we can state that control of pathogens can result in less disease and better welfare. But there are other components of welfare, and so usually the guiding principles for welfare just put 5 freedoms in the freedom from malnutrition and thirst, freedom from fear and distress, freedom from physical discomfort, freedom from pain, injury, and disease. We already have seen that, but also freedom to express normal patterns of behavior. And we'll see very rapidly that there are conflicting aspects between pathogen control and welfare, because most means of pathogen control decrease, in fact, the freedom of animals to express their normal behavior. For example, outdoor exploring, foraging, normal behavior with congeners. And a very good example just taken from humans with the example of COVID-19, with the confinement, obligation to wear a mask, vaccination, all these control measures were badly altered their lives and were badly accepted by a lot of people. So this was the first point, but the second point, conflicting aspect, is the question of stamping out. In many infectious diseases that are regulated, there is the stamping out of infected farm, for example, and it implies suffering and deaths of thousands of animals. And for example, with avian influenza, last year in France, in 4 months, we had to kill 1.3 million chickens. So in fact, the question is how we can balance between the positive and adverse effects of pathogen controls on welfare issues. and for the benefit of who? And this is not a very simple question. I'm not going to go in that questions today, but it is a question that we have to address every time we want to think about control measures. I would like also to stress out a little that a recent attention was paid by the veterinary profession for welfare aspects and consideration, because now they include both health and welfare in their approach. And I want to present a recent example about these antibiotic-free labels for poultry and pig. There is in fact a tendency by some farmers to refuse any antibiotic treatment, even for diseased animals, in order to keep the antibiotic-free label and a better price for that. And this, of course, has very bad health and welfare impact on the animals. So from that, there is currently a strong demand by veterinarians and also part of the animal sector for health and welfare monitoring tools and indicators on farm to better monitor the need and the use of antimicrobials. So this is in France. I think also it shares some in different countries. And maybe the same could arise for parasitic control as well. So now, if we move to human and social sciences point of view, in fact, it appears that there are different perceptions about what is health, what is welfare, what are the most important aspects to preserve between health and welfare if we have to balance. So what I presented so far is the vision of the veterinarian profession, whose main goal is to keep animals in good health. But in fact, there are other perceptions that depend on stakeholders. And even in the same profession, for example, for farmers, you can have different opinions on that. For example, for farmers, between those who prioritize the maintenance of good health for their flock, and they agree to have control measures of diseases, but others that emphasize the well-being of their animals and their freedom to express their natural behavior. So since I'm talking about farmers, I would like to tell a few words about sociological studies, current sociological studies that point out psychological distress of farmers that are facing this contradictory injunction, notably controlling disease on the one hand and ensuring well-being and freedom to express natural behavior of their animals on the other hand. And the main current example is that example of keeping poultry indoor to control avian influenza. And a lot of farmers do not agree with that. And just to continue and finish with these sociological aspects and highlight on the point of view of citizens and consumers, because they have a very huge impact on the future of farming activities for animals. So here I would like to tell a few words based on a paper from the UK by Clark and collaborators. This is a systematic review regarding public perceptions on production diseases and welfare. And I just pick a few points regarding our question today from their results. And I think this study was done with the analysis of 80 papers. So are more concerned by welfare considerations, females compared to male, young generations, and higher education people. And these citizens and consumers have quite holistic view of welfare and health, and they take into account both of them at the same time. And for them, for example, also what is bad for animals is ultimately bad for humans. So I come to the main point from that study that concerns what we're talking about today, is the question of naturalness. For citizen and consumer, naturalness is central in the relation to animals' behaviors and living conditions. and with enough space, freedom to behave according to natural instincts, and access outdoors. And because naturalness is central, it means that efforts to protect health at the expense of naturalness is not supported. So it means that citizens do not prioritize animal health compared to welfare conditions of the animals. So this is, for me, a very important point, because it can be in very direct opposition about the way we are working together and controlling animal diseases. So these allow me to come to a conclusion. So answering that question, does pathogen control have a positive impact on health and welfare, is not obvious. So just 4 points. So pathogen control has a very positive impact on animal health, but also on human health for zoonotic disease, and it remains a major mean to control infectious diseases. However, it implies constraints on animals that are prejudicial to their welfare regarding the freedom to move and freedom to exhibit their natural behavior. And depending on stakeholders, pathogen control should not be preferred at any cost, and there is a balance that needs to be analyzed. And I think this balance should depend on the dangerousness of the pathogens, the zoonotic risk, and the binding aspects of the control measures were needed to take into account. And so finally, societal concern tends to favor naturalness to the detriment of animalness. So for me, it means that we need a lot of justification, explanation, communication if we want to implement strong disease control measures. And that's done with this short presentation, and thank you for your attention. I would be very happy to have your comments and your point of view on this point, because there is not a single answer to that question, in my opinion. Thank you.
Thank you very much, Christian. So I'd encourage the audience to write questions in the chat if you have any questions for Christian. We have quite a few minutes that we can do some discussion here if you like. Christian, yeah, as you started speaking, I was writing notes to myself thinking about this avian flu thing and how it's such a conflict for people to bring animals inside. I was on a farm recently where the poultry had to be brought indoors because of the avian flu, but the housing wasn't conducive to good welfare at all. It was never designed for the animals to be housed continuously in this space. And I found it very concerning that I felt it was a huge price to be paid by the animals to control the flu, but it is a kind of a cost benefit situation. So do you think we could maybe reevaluate some of these infectious control measures in the same way as we look at experiments from a cost benefit point of view? You know,
I think so. And this should be done also considering the different points of view, the point of view animal, the point of view of the one farmer, the point of view of the farmers as a whole, and the society. And it's not a very simple question.
No.
And for me, it's very difficult. And I think we have to work also on alternative options, of course.
Yeah, exactly. Yeah. So I'm just wondering, have we any questions? I don't see any in the chat as of yet. I'm just checking that I'm looking at the right place. Am I right?
No questions in the chat.
No questions yet. Okay. Well, that was my main one. So, but if there's no questions, and if you have nothing you'd like to add, Christian, to your talk, we could move on to the next one, I suppose.
Oh, thank you.
Yeah, thank you very much again. Okay, so I'm going to hand you over briefly to Jordan Moore of Standard BioTools before Nicole gives her talk. Thanks, Jordan.
Fine, thank you. So let me just share my screen. Okay, so thank you for the, to just stop to my screen sharing. I'm sorry for that. I'll just start again. Okay.
It's fine. It's fine.
Can you see my screen okay?
Yes, we can see the screen.
Perfect. So I'd like to introduce some of Standard BioTools tools for helping with animal welfare and some of the genomics approaches that we can help you with. So obviously there are a lot of factors that we can't help with in terms of where the animals are going to be kept and the size of their enclosures and all of that, but we can help make sure that the conditions that they are in terms of their health are promoted ideally from a genomic standpoint. So that means looking at pathology of the animals themselves, detecting the pathogens, helping with marker-assisted selection and breeding to make sure that we have animals which are going to be the most resistant to the pathogens available, or presenting the best markers for living in the conditions that they are going to be in. Looking at genomic prediction and selection on that note, and then also looking at the microbiome, not only within the environment they're living, but also within the animals themselves. The way we can help with this is really reducing the costs of running these types of tests. And so therefore, we're going to be helping to accelerate the animal welfare and increasing yield and quality of the animals as well. Now, really where we're going to fit in the best is going to be in the quality control and pathogen detection, when we're looking at a smaller number of markers, as well as animal health and marker-assisted breeding. So we're really looking at markers from the tens to up to the hundreds and thousands. And we'll be doing this using our proprietary integrated fluidic circuits. If you've worked with these in the past, you know they're great of miniaturizing PCR and NGS preparation reactions. This means that we're reducing the volumes of those really expensive enzymes that you're normally using, and they will automate many of the steps. So this can eliminate the need for liquid handling. This can also eliminate the need of even having a large laboratory, because the instrumentation is quite small. Now, the workflow itself, and this workflow really works all the way across our gene expression and genotyping workflows, and gene expression that can even cover our pathogen detection, so looking at RNA and or DNA. You're going to run the extraction of your RNA or DNA. In this particular example, we're looking at DNA. The type of sample will not matter. We do want to have good quality DNA, but we can also use a preamplification step if the quality is poor, and we even integrated that into some automated workflows and automated IFCs, or integrated fluidic circuits. You design your PCR panels, and so these can be assays that you've already pre-designed. So these can be TACMAN assays, or assays that you're used to using with CyberGreen, or we can help you with the design from our side as well with our free D3 design service available on our website. You add your samples with a master mix to the integrated fluidic circuit. You add your primers and probes, or your primers, and those will be combined. And so, for example, on one of these IFCs, if you're running a 9696, this would be 96 samples, which you're testing against 96 assays. Those each sample will encounter each assay on the IFC. So that means that when we fill the IFC, we create a matrix. We're going to fill our sample chambers, then fill our assay chambers, and then we're going to allow them to mix. Once those have been mixed, then we're going to be testing 96 times 96. So that means on a single one of our IFCs, that's 9,216 PCR reactions. So your 96 samples will be tested for all of the 96 markers on the IFC. So all of the loading and mixing of the samples and assays, instead of having to combine those individually in a plate, are going to be combined automatically for you in the IFC. The table below here is giving you an idea of some of the different formats that we have. So the different numbers of samples and the different number of assays and the total processing time. And actually, we've even come out with some new workflows that are reducing this processing time. Once you're loading onto our X9 real-time PCR system, this can even be done overnight. So you can just run your load your assays and samples, put it into the X9 system, and your results will be ready in the morning. And you can analyze that quickly with the free software that's available on our website. It can be as little as 5 cents per data point, and we can even adapt for specific applications if you already have assays in a particular chemistry. So to give you an idea of the type of studies that have been done with our system, and there are way too many to be able to present in this very short amount of time. This particular one was looking at gene expression analysis of samples from the bovine reproductive system using our microfluidics platform. The DNA was extracted from 11 different sample types from the reproductive tract. A panel was designed for 96 genes, run on the IFC, put into the real-time system for PCR and data acquisition. And they were able to determine that this high throughput approach is not only reliable and as reliable, if not more so than a standard 96-well system, but it's an excellent tool for studying bovine reproductive systems, especially using quantitatively limited samples, since a large number of target genes can be assessed from a very low amount of starting material. To give you an idea, we're using 2.7 microliters or less per sample to look at up to 96 different markers. This particular study out of Korea was looking at improving animal breeding. So look at meat quality via single nucleotide polymorphism analysis, or SNPs. So this was DNA from muscle tissue from 192 Korean cows. The panel was designed for 3 different SNPs within the HSPV1 gene. Samples of that assays loaded to the IFC with the reagents necessary, then run for the real-time and the data acquisition, and results were analyzed. And so for them, using only one plate, 192 cattle steer were analyzed for heat shock protein beta 1 expression to identify DNA markers related to meat quality in Korean native steers. And these types of studies are very easy to implement. Really, what takes the most time is collecting your samples from the animals themselves. Some of these studies, just to give you a note, are done on our previous platforms in terms of real-time platforms, which have a multi-step approach. But the exact same, literally exact same IFCs and chemistries can now be run on the X9 system, which is much smaller and much more automated. So there's even less intervention from the individual technicians. In this particular study from the University of Illinois, looking at microbial research, this was DNA from fecal samples, of course, not really the greatest quality. There can be a lot of inhibitors at 4 different time points from 26 calves. And they looked at the microbial DNA by looking at a panel for the V3, V4 hypervariable region of the 16S gene. Again, samples were loaded onto the IFCs. This particular one was an IFC for library preparation, which can also be run on the X9 system. They ran the sequencing on the Illumina system, and they then analyzed those sequences. And they were able to use the low-quality DNA from calves and were able to identify potential changes in the microbiome and the metabolome that allow most efficient heifers to consume approximately 50% less solid feed. So that's what I have as a quick introduction to the type of studies that have been done on our system. If you are interested, of course, feel free to contact us or by myself or one of our other field application specialists, and we can give you more information on exactly how our technology works and can help you with your particular studies.
Thanks, Jordan. I just had a check in the chat just to see was there any quick question, but there wasn't. Oh yeah, there is actually. We'll just put this one quick question to you. Are there any regulations in Europe that prevent us from using this technology applied to animal diagnosis and QC?
That's a good question, and I have to admit that I don't know all of the regulations within Europe. So there may be some fields in which you cannot use it directly for QC. I would recommend that you contact us, and we can look into the particular cases that you're concerned about. But we do have many customers who are running this across many different fields, from even looking at the aquaculture to looking at the pathogens that are present within ticks, that to the pathogens that are going to be looked at within pork and other species. So I don't know the particular cases of each one, but we can look into that, and our specialists can find answers if there are restrictions. Of course, the animal space is less regulated than in the human space, but even in the human space, to give you an example, there are many laboratory developed tests that have been created using our technology, even for organ transplants from some individuals to others. So if the regulation doesn't allow it as a test that we've developed, because we haven't come out with a diagnostics type kit, in many cases it's possible to create a laboratory developed test and go through the validation yourselves. We even have customers who have gone through validation within France for solid tumor diagnosis, and that's using our technology. So even in the human field, it has been done.
Great, thanks, Jordan, and thanks for the question. If that's it, I think, yeah, thank you. Okay, I think that's it. So we'll thanks, Jordan. We'll move to Nicole Goka from Copenhagen University now next. Thank you.
Cool. Yes, thanks for the word, and thanks for giving me the opportunity to present my work here. My presentation will be about the design and validation of different high throughput real-time PCR systems that can be used for detection of porcine and bovine respiratory and enteric pathogens. And to begin with, I will give you a short introduction to some thoughts and ideas behind the use of this high throughput platform. Oh, sorry. Yes, in 2015, I started as a PhD student at the National Veterinary Institute in Denmark. And at that time, this institute had a large diagnostic unit where the main focus was on diseases of domestic animals. Each year, this institute thousands of samples from different animal species, such as pigs, cows, birds, and minks. And all these samples were tested and analyzed with the aim of identifying cause of disease and to identify known and unknown pathogens in the Danish animal. And furthermore, the institute also conducted surveillance program of avian and swine influenza A viruses. And the sample flow at the institute was as follows. So the samples were often taken from deceased animals, and then the samples were submitted by the veterinarian to the institute, where the samples were analyzed for the presence of pathogens based on the request from the veterinarian. And then the test result was sent to the veterinarian, who could then determine a specific treatment for the animals based on the laboratory findings, which then hopefully led to a healthy animal and a happy farmer. And one of the technology used for this pathogen detection was the PCR technology. And at the institute, they used a so-called low-throughput real-time PCR platform that has a sample capacity of either 36, 72, or 100 samples, and the samples could either be analyzed in one PCR assays or up to 6 different PCR assays in the same run, depending on if the assays were running as singleplex or multiplex. And at that time, most of the assays were running as singleplex. When using this low throughput real-time PCR platform, the analysis cost per sample is quite high due to the use of expensive reagent, and this high cost per samples had led to the many farmers keep their sample submission to a minimum. And reduction of sample submission also means reduction in laboratory analysis, which leads to that fewer animals get diagnosed. And lack of analysis can result in less specific treatment because the veterinarian does not have the right basis for the treatment. And therefore, there's a risk of that medication and vaccination will be based on clinical observation instead of laboratory investigation, which can lead to the risk of overuse of antibiotics and suboptimal vaccine programs. So in order to address these challenges, my PhD project was started. And the main aim of this project was to develop different high throughput real-time PCR systems that could be used for detection of and typing of pathogens. And here I'm talking about viruses and bacteria. And the idea with this system was that they should be specific for pathogens which are important to the health and welfare of the Danish pigs and cattle. And furthermore, a purpose was also to reduce the analysis cost per sample. So we wanted to change from this so-called low throughput platform to a high throughput platform. And we decided to work with this high throughput real-time PCR platform called Biomark from Fluidigm, or now Standard BioTool. And this is a system that use less sample and reagent compared to a traditional real-time PCR platform. And with this system, you can provide above 9,000 reactions in one run. And it is a chip-based system, and there exist different chips format where you can see some of them here to the right. And the choice of chip depends on how many samples and assays you have. And here you see the overall workflow for this high throughput procedure. So first you have to extract the RNA and DNA from the samples, and then you have to pre-amplify the samples. And the reason why you have to do this pre-amplification is because the sample volume used on the chip is very small. So you pre-amplify your samples in order to obtain more copies of your target. And after the pre-amplification, you add the pre-amplified samples to the chip together with the PCR assays, and then you load the chip in a so-called controller where the assays and samples are mixed. And after that, you place the chip in the Biomark, which is the real-time PCR machine, and then you run the PCR. So one of the high throughput system we have developed is a system we just call the porcine high throughput real-time PCR system. And it is a system that can be used for detection of different viruses and bacteria. And the idea with this system is that this system is contain PCR assay specific for all the relevant pathogens known to cause respiratory and intestinal disease in pigs. So when using this system, you reduce the risk of not detecting the relevant pathogen or pathogens. So here you can see which pathogens that are included in this porcine high throughput system. So here we both have respiratory pathogens and enteric pathogens. And a very good thing with this Biomark platform is that you easily can add and remove PCR assays. You just have to ensure that the PCR assays can be run under the same PCR conditions. Furthermore, we have also developed a high throughput system that can be used for subtyping of influenza A viruses in pigs, meaning that this system contains PCR assays specific for all the different subtypes known to infect Danish and European pigs. So after we have designed and developed this porcine system, we wanted to test the performance of the system on field samples. So therefore we conducted a study with 10 Danish production units. And in this study, we also wanted to test if monthly monitoring of pathogens on herd level could be a supportive tool for the veterinarian. And this monitoring of pathogens should be combined with observation of clinical signs. So this study was carried out in the 10 pig production units units, where 6 of them were nursery units and 4 of them were finisher units. And the sampling and clinical observation were carried out in each unit 3 times, namely in September, October, and November within the same year. And at each sampling, 3 different age groups in each herd were selected. Within each age group, 2 pens were sampled as shown here in the figure. And the sample material we collected were fecal suck samples and oil fluid samples. And the fecal suck samples were obtained by having a suck on the outside of the boots, and then you walk around in the pen for a while. While the oil fluid samples were collected by hanging a rope in the pen for approximately 30 minutes, and then the pigs were able to chew in it. Furthermore, at each sampling, clinical signs as coughing and diarrhea were observed and registered, and based on these clinical observations, so-called coughing event and the diuretic event were calculated. So in total, we collected 174 fecal suck samples and 172 oil fluid samples, and all these samples were then analyzed on our porcine high throughput system. And in this system, we found that, in this study, we found that there was a significant correlation between the presence of the viruses, influenza A virus and porcine cytomegalovirus, and a coughing event if these 2 viruses were present with CQ value below 20. Furthermore, we also found that there was a significant correlation between the presence of the bacteria Brachyspira pilisocoli and Lawsonia intracellularis and a diuretic event if these 2 bacteria were present with CQ value below 26 and 24 respectively. So based on the experience we obtained in the study I just told you about, a new concept called SOS was started up in Denmark, and this SOS stands for Swine Objective Surveillance. And this concept is supposed to be a health monitoring system. And the concept of SOS is based on somatic sampling of oil fluid and fecal suck samples in both the farrowing, the nursery, and the finisher units. And the samples are sampled at a regular interval throughout the year, 4 to 8 times, and there are common guidelines for all the herds, and it is the farmer that are responsible for the sampling. And after each sampling, the samples are then sent to University of Copenhagen, where we analyze the sample on our porcine high throughput system. And then a report with the result are then sent to the farmer and the herd veterinarian. This SOS concept has been running in Denmark since February 2020, and so far 42 herds has been using this system. And we have got some feedback from the veterinarians and farmers, and some of it sounds like it gives an informative overview of pathogens in each age group. It is a really good health check. We can be one step ahead. Our use of antibiotic and vaccines are correct and sufficient. So this SOS concept has showed us when using this high throughput platform for monthly monitoring of pathogens, we obtained a lot of information about. We obtained a lot of information within each herd, and this information can be used to see which pathogens that circulate in the different herds. Furthermore, this knowledge of the pathogen dynamic in a healthy status can be helpful in an outbreak situation, since the farmer and the veterinarian already knows which pathogens that are present and in which level. And this greater knowledge about the pathogen pattern within a herd can hopefully lead to a more specific treatment of the animals. So beside the different porcine system, we have also developed a bovine high throughput real-time PCR system, and this system can be used for detection of different viruses and bacteria. And again, all the relevant pathogens known to cause respiratory and intestinal disease in calves are included. And here to the right, you can see which viruses and bacteria that are included in the system at the moment. And this bovine system was developed in a big calf project, which was called Robust Calf. And this project was running in Denmark in 2018 and until 2021. And in this study, samples were collected from 100 Danish herds, where 83 of them were dairy herds and 17 were slaughter herds. And within the dairy herds, 3 different age groups were sampled, while in the slaughter herds, 2 different age groups were sampled. And the samples were collected in 2 periods, namely in September to April in 2018 and until 19, and again in the same months 1 year after. And then at each herd visit, they also did clinical observation where they looked at different parameters such as rectal temperature, coughing, nasal and eye discharge, diarrhea status, and so on. So this figure shows the result for all the samples that were collected at the first visit in the 100 herds, and all these samples were analyzed in pools. And this figure shows the result both for the nasal swab pools and the feces pools for the different age group in both the dairy and in the slaughter herds. And looking at the result for the nasal swab pools, we found that bacteria were more frequent than the viruses, both in the dairy and in the slaughter calves. And for most of the bacteria and viruses, the highest prevalence was found in the oldest age group. And looking at the result for the feces pool, we here saw that the viruses were found to be more frequent than the bacteria, with rotavirus A being the most prevalent one, followed by the bovine coronavirus. One of the interesting findings here was that we found that some of the pools tested positive for influenza D virus. And I think it is an interesting finding since this is a relative new virus. This virus was detected for the first time in 2011 in the USA. And this virus has not been described in Denmark before. So therefore, we were interested in to look more into these positive findings. And looking at the phylogeny of the glycoprotein of this influenza D viruses, you can see that there exist 4 main clusters. So we were interested in to see in which cluster or clusters the Danish sequence belong to. So therefore, we sequence all the positive influenza D samples using next generation sequencing, and here we found that the Danish sequence clustered within 2 different clusters, namely the DOK and the D660. An interesting thing here was that all the samples collected in 2019 clustered together in the DOK cluster, while the samples collected in 2020 clustered together in the D660 cluster, except for 1 sample. Furthermore, we also sequenced a sample, an older sample that had been collected in 2015, but this sample was first recently analyzed for influenza D. But the finding of influenza D in this sample could indicate that this virus actually have been present in the Danish calf for several years. So last year we ran another Robust Calf project called Robust Calf 2022, and this was a 1-year project. And the overall aim of this project was to monitor disease-causing pathogens at herd level in 6 Danish slaughter herds. And the idea here was to collect samples in the 6 herds 4 to 5 times, and then analyze all these samples on our bovine high throughput system. So this setup in this project was meant to be like the SOS project eller concept. And the samples that were collected within this project were nasal swab samples and fecal samples. And the nasal swab samples were collected from 5 calves from 3 age group. And these age group were defined as the last arrived calf from the group that were most recently weaned and mixed together, and calves that were approximately 3 months old, while the fecal samples were only collected from the youngest age group. And the samples were collected every 2nd to 3rd months by 3 veterinarians from 3 different companies. And each of these veterinarian has selected 2 of their slaughter herds. And 2 of the veterinarian collected sample 4 times, while the 3rd veterinarian collected samples 3 times. So, and then all these samples were sent to us, and then we analyzed them on our bovine high throughput system. And after each sampling round, we made a report containing the result for the samples at the sampling round. But this report also contained a comparison of the result between the different sampling rounds. And here you can see an example where to the left you have the result for sampling round 1, and to the right you have the result for the sampling round 2, and then you have a table per each group. And this was just to give the veterinarian and the farmer an overview of which pathogens we have found within the different age group and within the different sampling rounds. And this table shows the result for all the nasal swab samples collected in this project for all the 6 herds and for all the different age group. And the numbers in the squares are the number of positive calves within a certain sampling round. And looking at these results, we found that some of the pathogen, here some of the bacteria were found to be present in almost, were found to be present in all the sampling rounds and in almost all, in almost all the animals, while others pathogens, especially the viruses, were not detected that often. When the veterinarian of herd number 5 and 6 received these BRSV positive results, he wondered because none of these calves showed a symptom of being infected with this virus. But then he told me that within these 2 herds, they vaccinated against this virus using live modified vaccine. So therefore, we decided to sequence all the positive BRSV samples and also sequence these 2 vaccines in order to see if these PCR findings could be due to the vaccination. And in herd number 5, they use a vaccine called Bovilis ASP live vaccine, while in herd 6 they use a vaccine called Nassim vaccine. But for both herd, we found that there was a high similarity between the vaccine strain and the strain we found in the positive samples. And the company, the Bovilis company, also claimed that vaccinated calves can excrete the vaccine strains for up to 12 days after vaccination. So it is a thing you have to be aware of when using these live modified vaccines. In 2 other herds, we made a minor study where the veterinarian, beside the nasal swab and the fecal sample, also collected nasal wipe wipe samples, and these nasal wipe samples were collected from the same animals where he also collected the nasal swab samples from. And here we wanted to look into and here we wanted to see, we wanted to compare the pathogen findings in these two sample materials. And in this table, you can see the results for the nasal swab samples and the nasal wipe samples for sampling round 4 and for sampling round 5. And as you can see, there is a good correlation between the pathogens we found in these two sample materials. An interesting finding here was that in sampling round 4, all the animals tested negative for influenza D virus, while in sampling round 5, all the animals tested positive for this virus. And then the veterinarian told me that shortly before the sampling, there was a severe disease outbreak in this herd with increased mortality. And on the basis of the symptoms that the calf showed, it could well be the influenza D virus, which was the cause of this disease outbreak. Yes, then I just want to thank all the different people who have contributed to the project presented here. And and then I would like to thank you for your attention.
Thank you very much, Nicole. A lot of work done, very productive work, a lot of publications. Thanks for that. There's been a few types of housekeeping queries, and there's one on the cost comparison per sample using the chip PCRs. And this person wants to know, does the sensitivity compare? So first of all, it's the cost comparison.
Between the two platforms,
I guess, yeah. What is the cost comparison per sample using the chip PCRs? And does the sensitivity compare well? I presume with traditional measures.
Yeah, so I can start with the sensitivity. So in our validation of these different systems, we have run a lot of comparison between these two platforms, and the sensitivity is quite the same. It depends assay from assay, but overall it is the same. And about the cost, you can say that the cost the cost is the same between these two platforms. So using this low throughput platform, you can get one answer, while you can get up to 24 answers using the high throughput for the same cost.
Wow. Good, that sounds compelling. Thank you, Nicole. Okay, now there's just thank you for your presentation from some of the people in the audience. And we don't have any other queries. So if anyone has queries either for Nicole or any of the other speakers, this is coming up to your last opportunity. Otherwise, I just like to remind everyone again of, the details on these EAP webinars are on the website, and the EAP YouTube channel will host the recordings. The next webinar is on the 14th of March. Work like an ant, collaborate like a bee, insect research collaboration initiatives in Europe. If that's it, I think it's just up to me to say thank you to all the participants and to all of you for attending, and to the support for me from the EAP to chair this and host the event today. So thank you very much, everybody, and have a nice afternoon.